Hi, I'm Scott Coyle, and I'm going to be your instructor for Section 4 of Biochemistry 501, in which we're going to be learning about nucleic acid biochemistry and protein synthesis, among other interesting topics. Now, I just want to let you guys know that this is my first time teaching this section of the course, and so my goal is to do as good a job as possible, but I also want you guys to know that if there are components of the presentation or the way that topics are covered where you guys feel that you need more clarification or there are modifications to how I'm doing things, just feel free to let me know, because my goal here is to create as clear and helpful a set of videos and lectures for you so that you guys can achieve total mastery of this important material. I'll add that in addition to the core lecture videos and slides that I'll be posting, I plan to make accessory videos that will help you see how to go through certain problems that you might encounter on problem sets or on exams. I'll emphasize that there won't be any new content in those lectures. That is, you don't need to watch those accessory videos in order to be able to do everything and know everything you need for the course. But I'm hoping that through these videos, we'll get an sort of additional kind of experience that could be helpful for you guys studying and learning the material. And so if any of you have suggestions or ideas for things that you'd like me to go through with respect to that, please let me know, and I'll be happy to try to tailor those videos and the accessory content to things that work well for you guys. And so with that, I thought we could just dive in and sort of give an outline for what we're going to be learning about in part 4 of the course, which is going to be largely focused on genetic information transfer. And so to motivate what this section is all about, let's quickly review what you've learned in this course so far about the biochemical basis of life. You learned what proteins are. You learned that there are these fantastic polypeptide chains that can fold into complex three-dimensional structures, and those structures that those proteins adopt enable them to function as enzymes that can catalyze a whole slew of important and distinct chemical reactions inside the cell. You then learned that by taking different enzymes and connecting them together in the cell can build chemical pathways that can do all sorts of interesting things. They can harvest energy from chemical compounds like glucose, or they can extract the energy of light and store it in things like ATP. You also learned how cells can build new things using chemical pathways, such as the building blocks of most living systems like amino acids, lipids, and so on and so forth. And so at this stage, you have a pretty good handle on sort of what the moving parts are inside of a living cell. But an interesting question comes from asking, where does the information to make all of those enzymes come from? How is that information stored at a molecular level? How is it actually used to specify what proteins it should make? How does some sort of molecular information specify a protein sequence? And how is that information actually passed down from one cell to the next, so that every time a cell divides, the information needed to make all of those pathways and enzymes that you've learned about in the earlier part of the course, every cell has that information. And so in this section of the course, section 4, we're going to be learning about the answers to all of these questions and more. And so the key outline for this section of the course is to understand the molecular basis of genetic information transfer. And importantly, you will develop a mastery over what is known as the central dogma of molecular biology, which is that DNA encodes genetic material which can be replicated. DNA can be used to template the synthesis of an intermediate molecule called mRNA, and that mRNA encodes can be translated into protein sequences based on its sequence itself. And it's going to be really critical when you walk away from this course to understand the central dogma of molecular biology, because it's the foundation to be able to understand most cell biological or organismal scale questions. It's also critical to be able to understand and interpret the mechanistic origin of many diseases. And finally, it is a powerful technology that our understanding of has enabled us to modify, engineer, and take control of living systems to repurpose them to do new and interesting things. Now, as we dive deeper into this material, you will learn that the genetic information that codes for proteins is contained in nucleic acids. And there's really two types of nucleic acids you're going to learn about. Deoxyribonucleic acid, or DNA, which I'm sure everyone in the class has heard of. This material is the master information store of genes that encode proteins. It is what is replicated and passed down from one generation to the next. So this is the master store of the genetic information needed to make all the proteins inside the cell. You'll also learn about ribonucleic acid, or RNA, which acts as an intermediate messenger that is transcribed from DNA and provides the templates, which we call messenger RNAs or mRNAs, for the synthesis of proteins in a process called translation. So these RNAs are basically an intermediate that is used to make protein. You will learn about the structure of DNA and RNA and how these structures that these nucleic acids adopt facilitates their role as being an information carrier in the cell. This will make it clear to you why nucleic acids are so useful for this process of replicating and transmitting information. You will learn key biochemical features of these molecules that will provide you with a logic with which to understand every core molecular biological process, replication, transcription, and translation that goes on inside the cell. And in addition, you will learn how the integrity of the DNA in the cell is maintained through quality control and repair mechanisms. Because obviously, if this is your master store of genetic information, you need to make sure that it's well taken care of, otherwise problems can arise. To actually access the information of nucleic acids and convert them into protein, you will learn about the enzymes that use genetic information of DNA to transcribe messenger RNAs. So these are called RNA polymerases. You will also learn about the enzymes that translate the information stored in an mRNA into a protein. So how do we go from a nucleic acid to a protein? That's going to occur through a process called translation that you'll learn about as well. And finally, you will learn about the mechanisms with which the cell uses to regulate which genes are transcribed and translated at different times, and how the cell can use that regulation to dynamically change and adapt its state to different conditions. So for example, in your body, there are different cell types, and different cell types are expressing different sets of genes. How is that achieved? We are going to learn in this section of the course. And then finally, we're going to learn a little bit about how understanding all of these processes, nucleic acid biochemistry, replication, transcription, translation, that has given us a foundation for laboratory techniques and powerful emerging technologies that are going to have a huge impact on society. So you may not go on to be a scientist, you might go and do something else in your life, but if you understand this material, then you're going to be well equipped to understand a lot of the pressing issues that are coming in society with respect to these technologies. So you'll learn about cloning, the process of taking genes out of their native context, cloning them into systems where they can be studied, and their gene products investigated in the laboratory. And we'll learn about how everything that you've learned in the course will prepare you to understand how CRISPR-Cas9, a technology that I'm sure you've all heard of, can be used to modify the genome of almost any living cell. And finally, how tools like CRISPR and cloning can be used to engineer cells to express new proteins and rewire behavior. And we'll go through some examples of how that's been used to treat diseases. And so with that overview of the course, let's get started on our first lecture going into the actual details of this material.